So my last video got cut off, so this is the part two of my biotechnology project on DNA sequencing. I'm going to pick up where I left off on natural DNA replication compared to the DNA sequencing process. So for natural DNA replication, the enzymes DNA helicase and topoisomerase work together to unwind DNA strands. By cutting the strands and breaking hydrogen bonds so that the nitrogen bonds are able to link together. And once the nitrogen bases are exposed on a DNA strand, synthesis of the DNA sequence can commence. Compared to the DNA sequencing process that uses serial analysis of gene expression, which can allow researchers to determine specific genes that are expressed in a sequence and how prominent they might be. And so, by scientists using a large quantity of DNA in a polymerase chain reaction, they can generate many copies of the same genetic sequence at a very fast rate, even faster than natural DNA replication can do. And then, so for the second point, DNA polymerase 3 initiates the synthesis of DNA strands in natural replication, whereas in the DNA sequencing, a purified DNA polymerase, which is the primer, is what synthesizes the DNA strands of the various lengths. And then for the third point, both the natural replication of DNA and the DNA sequencing process is created in the 5' to 3' direction. And then this little side point is one of the strands moves towards the replication fork and the other strand moves away, which is what is able to maintain the helical shape. For the fourth point, the RNA primer temporarily binds to the 3' end of the parent DNA strand, so nucleotides can be added to the existing strand of DNA as it continues in the natural replication process. Whereas in the DNA sequencing, the dideoxynucleotide triphosphates are terminating bases because it lacks the 3' hydroxyl group. I already mentioned this in one of the previous slides. But that's the hydroxyl group is what is needed to form the phosphodiester bond between one nucleotide and the next. So that's what cuts it off. And then lastly, for natural replication, DNA polymerase 1 is what proofreads each DNA strand to make sure that it has the correct sequencing. And if there is an error, it can remove the error before it has the chance to be passed on to more cells. And if the error of the DNA synthesis isn't fixed in time, that's what leads to the mutations, because before the DNA polymerase 1 can delete the error, it already is passed on. And then for DNA sequencing, it's the DNA is transferred and organized through the capillary gel electrophoresis to ensure that each gene is in primal condition, and then the hope that there would be no mutations present, which is why people would use the DNA sequencing process. Like, for example, in the CRISPR technology, this is what they would use. And lastly, I'm just going to share my opinion on how I feel about DNA sequencing. And overall, it has been a really promising and successful field that a lot of development has come out of it. And I believe that research should continue on finding the treatments for the diseases and mutations, as well as using the genome sequencing to find different never before known mutations. And so that people are less susceptible to contract more diseases, because we don't want people to be sick all the time. But the area of DNA sequencing that should halt, in my opinion, is the CRISPR technique. The genetically modifying the embryos, I think, is not a good thing to do, because you're changing the specific mental and physical characteristics and adjusting the baby's gene sequence before it's even implanted in utero. And I think that is completely wrong and not a very good way to approach the whole situation. The reproduction, it's not meant to create the perfect baby where you want it to be able to be very smart and athletic and not have any mutations, but you can't just create a child without any predispositions to illnesses or potential behaviors, because that is not the whole purpose of reproduction. And if you really wanted to have that baby, you would want it to have characteristics that are found in yourself, because that's what the baby is supposed to be about. It's supposed to be the creation of you and someone else and making it have similar attributes that you have. So the CRISPR technique would not allow this to happen, and that is why I think that the research for this area should stop. And that is the end of my presentation of DNA sequencing. I hope you enjoyed it and that it wasn't too long and boring. And thank you so much for listening.